Okay. And this to go. Okay, well, thank you very much for inviting me to give the seminar here, and it's a pleasure to be able to talk to you and show a little bit of our work on multiphoton imaging in cancer biopsies. So our place is a lab in the physics department of Federal University of Minas Gerais, and is it okay? Is it better now? And I will just start by showing where we are. So that's Brazil in South America. And the state of Minas Gerais is this, so I don't have the pointer there, it's just this part in the inland there. Can I try to put the mouse here? Just let me see if I can. Yeah, I can do it with the mouse. Yeah, it's better. So that's the state of Minas Gerais is this part of Brazil, which is in more detail here. We can see it is north of São Paulo, which is much well known, and west of Rio. We are inland in the mountain areas of the this region of Brazil. And that's a photo of the campus in UFMG, which is in the part of the city which is called Pampulha. It's to the north of the city. And so this area here is the campus there. So our building is this one in this part here. So we have a central area, which is the area of facilities with restaurant, which is very close here. And in this same, in the region around here is the veterinary school, where they have a hospital. And so part of the work that I'm going to be showing is with some collaboration with people from there as well. And that's the more close picture of how our building is. This is the physics building, one of the entrances, and that's the side part of it, which is that facility central area. And then I'm going to show inside the building. That's a photo of part of my group, which is just in a garden. Our building has some open corridors with internal gardens, and this garden is just in front of our lab. And then as I'm talking about the people, I'll show who are the collaborators beforehand. And so those are the students in my group working with this cancer biopsies imaging. So an undergraduate students and 3 PhD students that did their work in this area of research. And then the collaborations, because of course we do just the physics imaging part of the work. And the other part is together with the, one of the work is going to be in mammary cancer in dogs, which is together with the people from the Biology Institute and the veterinary hospital. The other one, which is in prostate cancer, is together with the pathologist, which is Eduardo Paulino Jr. and Marcelo Mamedi from the Faculty of Medicine in UFMG. The Faculty of Medicine is not in the same campus, it's in the city center. And so it's a little bit of trips around the city with the samples to do the measurements. And the image analysis, which is a strong part of this work, was a work done together with the Frank Longford, which was a postdoc that stayed a few months in our group in Brazil, and the University of Southampton in UK. So now I'm going to start to say what is the outline of my talk. We are doing mainly the multiphoton optical microscopy. I'm going to go in details what this multiphoton means, but we use the idea of scanning image, where we have just a laser, which we use is a femtosecond laser, scanning the area of the sample that we wanted to measure. And we measure point by point on that sample the processes that we want to look at the material. And one of them is second harmonic generation. I will show a little bit what it is. And the other one is two-photon excitation fluorescence that we use to many biological materials to characterize how these processes change it point by point in the sample. And in this talk, I'm going to be showing details on cancer biopsies. And so the main motivation, for this audience is easy to say, but it is the cancer is the second leading cause of death globally. And two important ones are breast cancer, mainly in women, and prostate cancer for men. And so those are the two types that we studied mainly, well, not exactly breast, but close to breast. And the standard diagnosis, the end diagnosis is still based on visual inspection of biopsy, histological images. That's one of them. I know I'm not going to be talking about the immunohistochemic classification here, but that is one of the standards that is really hard work for the pathologists because it is very intense to really diagnose in detail many samples. And so I know from our colleagues there that they say that it's very hard because it's influenced by subjectivity, and it depends a lot on the specific training of the pathologists for each specific type of cancer. And so what is very desired is just to have a more quantitative way of measuring the biopsy and saying which type of cancer it is. So the example here is just showing that that's an example of the prostate biopsy colored with hematoxylin and eosin. And the hard task of the pathologist is just to go in details of how this cell changes and how is the tissue organization, and from that to characterize which degree of cancer or which type of cancer it is. So our work is to try to get some parameters looking at imagings to be able to help the pathologist with that task. So what is the scanning microscopy? Is the idea as is done in all confocal microscopy. You have a laser point that you scan with a mirror through an area of the sample. So you scan X and Y, and you acquire an intensity image of the whatever process you want to look at the material point by point. And so in our work, what we use is, instead of just using a continuous laser, which is a continuous intensity in time, and for biological applications, you need to use low power, otherwise you damage your material. What we use is a pulsed laser with very short pulse duration on the order of femtoseconds, so that we can concentrate all the energy of the laser in a very short time. And so there are some numbers here, but if we compare, for a laser with 100 megapulses per second, 100 megahertz repetition rate, we can, with a femtosecond pulse, have with the same average 1 milliwatt power, have a pulse, a power per pulse, which is on the order of 100 watts, or say. That's 5 orders of magnitude higher as compared with the average here. And with that, we can drive a lot of nonlinear processes in the materials, because the electric field on these very intense pulses just perturbs very intensively the polarization or the absorption in the samples. And so we can do a lot of nonlinear techniques, which is very easy to characterize details of the materials. One of the examples here is just looking at fluorescence. Normal fluorescence, as like in confocal microscopy, what you have is that the material absorbs one photon and emits another color, which is characteristics of, say, of the dye that you are putting to mark the molecule or the cell. But that's a process that happens all the way through the path of the laser. When we focus a laser in a material, what we have is something like that. It's a very small area where you concentrate the laser intensity. And for linear processes, the sample can absorb all along the path through the focus. As shown in this image here, that's an image just showing a continuous laser in a cell, in a cuvette with a dye. So we can see the fluorescence all along the laser path until the focus. But if we have a very intense pulse, we can have 2 photon absorptions instead of this process happening with just 1 photon. The material absorbs 2 photons at the same time. It makes the same transition, and then it fluoresces. With that, the process depends on the square intensity of the laser field, and so it happens just on this very tight part of the focus. And so it can be much more localized in position in the sample. And so, and we can see deep inside in scattering tissues. So that allows to do quite nice 3D resolution imaging. And because we use infrared lasers, it scatters less in the tissue also. And so we can do it very deep inside the materials. And these processes started to be used quite a lot in vivo imaging and biological tissues since around the 2000s, when these femtosecond lasers started to be more commercially available and more used from other fields, except from just physical labs. So the other, to do that, what we do is just we attach the laser, which is a Thai sapphire laser, is that pulsed femtosecond laser, in a scanning microscope, which is same idea as the confocal microscope. And we do it just, you put the laser through those scanning mirrors and scan the area of the sample that we want to do. And we collect the fluorescence on backscattering, and we can image them with different filters in different ranges of emission, so we can separate different, say, proteins or different molecules in the observed tissue. And with the same microscope, we modified it to include an extra detector just on top of the objective that is used to do the second harmonic generation. Second harmonic generation is a nonlinear polarization of the material that allows you to, the materials with non-centrosymmetric properties, which is like the collagen, say, in many of which is common in most of the tissues. It interacts with the laser. It's like in music. If you stretch strong the chord of a violin, you have the harmonics of the sound. And in the material is the same idea. So you get out with a wave which is double the frequency from the inputting wave. And so just specific materials like the collagen, for instance, in the biological tissues, can generate this second harmonic signal. And so it is very specific, and you can map very well in the tissue what is collagen and what is not. And so in the microscope, what we did is just with that extra detector, we put it just on top of the objective. And so we can collect in the same position of the sample, the fluorescence, the second harmonic, and it's not shown here, but we can collect also the transmitted image through the biopsies. And so that is just to show an example. That is the intensity map of the second harmonic generation through a prostate biopsy. And this is a 3D image that's been just compressed to our 1D, just to show how we can do. And this area is 700 by 700 microns that we could image the second harmonic through 100 micrometers in depth. And that is done in a fresh cut, it was just a punch biopsy with no treatment, nothing at all, just in the same formalin solution that it was taken out. And so we can image it without no processing at all and get the details of the 3D collagen organization. And so from these measurements, it was already quite few results in the literature showing that the organization and details of alignment and behavior of how it is stretched or waveness. And so there are a few results in the literature showing that that changes quite a lot with the degree of evolution of the cancer. So now our first results, we just had the prostate tissues, and it's blocking here, but I'm just showing two examples. That is just a hematoxylin and eosin.
Move this one. No, sorry. Okay.
Okay, thank you. Yeah, let's go back a little bit then. Yeah, thank you. And so the first results in the literature in this area was showing mainly that the collagen organization around the glands in areas of different type of cancers were being organized and stretched, and amount of collagen was also changing. And so the first ideas in the literature, there are a few papers since about the last 10 years showing that depending on the cancer, you get different organization of the collagen around the glands on the tissue. And so this first example that we did is just a prostate cancer. The top one is just the normal tissue in a HE cut. The same idea as is used by the pathologist to do the visual histological diagnosis. And we image it with the second harmonic intensity. So this green image here is just the intensity of the second harmonic on the same position of this HE microscope image. And that's an example of a normal healthy tissue. And this one is one type of the prostate diagnosis that I'm going to be going in more details later on, which is this pattern which is called Gleason 3. And from the image, you can already see that the details of the collagen is quite different. But what is used in the literature is just to try to go through this image and try to measure how these fiber lines are organized. And we can see that here is an example of patterns of prostate cancer with 3 different types of diagnosis. And the red line there is indication on how organized the fibers is. So the smaller, the length of the line shows the alignment of the collagen fibers. So this long line means that this area marked by this yellow square has collagen, which is much more aligned than this one. So those are the main results that were shown in the literature up to 2015, 2016-ish. And we could see that just measuring that in a number of samples, we can get, say, an average alignment property of the collagens that really shows that those alignment increases with the advance of the cancer. And so this type of works were very common in the literature for many different types of cancer. Breast, ovarian, and pancreas, many different types. But people were just showing that, well, if we look at the collagen, we can see it's getting, in some of them, it gets much more aligned. In some, they kind of just make very stretched lines around the tumor, but there was not any more specific correlation on how that can compare with the details of a diagnosis in cancer. And so when we look at that, we decided that, well, the idea to go forward and try to get that into a more practical way of diagnosis is to try to study that, the overall tissue organization, not only look at the collagen, but try to see if we can, with these images, extract a bit more information of the gland shape as well, and all the details that are really the main important one that the pathologist needs to pay attention when they are screening all the types of cancer. And so we decided to do, as I showed in the setup before, in the same position, try to get the second harmonic image, the 2-photon absorption. The 2-photon absorption fluorescence gives more information on how the glands and how the cells in the glands are organized in the tissue. And with the transmission image, we can get an idea also which parts of the cell are transmitting more the laser or not. That gives also a little bit of information on the overall tissue organization. And then we started, although we can do it in fresh biopsies, we started with the same histological biopsies, which is exactly the same ones that the pathologist was using to do the normal diagnosis, for us to get a very good direct comparison. And then we developed an automated image analysis software to be able to acquire quantitative parameters to get a method of diagnosis. And so the first work I'm going to be showing here to you is this one, which were in canine mammary cancer. And it's interesting because from our colleagues, they tell us that it's a very good comparative model to breast cancer, and it has some interesting practical ways of working as well, because we just use the normal natural evolution cancer in dogs. They were the dogs that were being treated in the hospital, the veterinary hospital. And so we didn't need to induce any cancer. It was just the animals that were being treated at the veterinary hospital. And so what we did, we separated a number of different cancer in dogs to compare what we can get with the second harmonic and 2-photon imaging and the diagnosis from the pathologist. And so our work is that we have this HE image, which is the one that is diagnosed by the pathologist. And so the pathologist selected the areas which were typical for each type of cancer. And we did a second 2-photon fluorescence image, which is colored here in red. The second harmonic image, exactly on the same position, which is the green intensity map here. And so that's an example of just overlaying the two together, so we can see that it matches well the areas of collagen and areas of cell components of the image. So we did that for a range of samples. So we started 55 female dogs with 416 histological biopsies regions. And in this work, I'm going to show just these 3 types of histological cancer, which is called the in situ, the mixed carcinoma in mixed tumor, and solid carcinoma. So those are the examples of the HE biopsy areas. In this second line, I'm showing the second harmonic intensity image on the same area and the photoluminescence image in the same position. So with that, what we did is then we took the images and we got the second harmonic, the fluorescence, and the transmission images were used to make a composite RGB image to be treated by the software. With that, we could get an automated image analysis to extract the fiber network in the image and the cell regions in the image automatically. Because the main task is that if we're going to be using that as quantitative measurements, where in that image are we going to measure what? And so we wanted to be able to automatically get the images that we take from the microscope and do treatments in a very large amount of images to have enough statistics in an automatic way. And so those, we used some techniques of image analysis that transform that image in a tensor. And then with that, we can calculate intensities and many details of the processes in each point of the image. So what we can get from the software is this idea. We can get a segmentation of fiber and cell regions in the image that are shown here in different colors. So this first line is showing the collagen network of fibers extracted from the images. So we are showing here in different colors in this image the collagen network of fibers that are connected together. So each collagen network is shown in a different color. So say in this cellular matrix in this area, which is well and nicely connected. And then within the gland area, we have smaller networks, but they are well-behaved network because the cellular regions there is just the normal health tissue. When we go to the different types of cancer, we can see that especially inside the gland, in many of them, we lose a lot of the collagen because you've got the cell proliferation, and the type of connection is quite different also. And so that is a feature that we can try to look and we're using that to characterize the details of the evolution of the cancer. In the second line, also in different colors, are shown the areas of cellular identified by the software. And so say in this one, we see this big red area, which is the internal part of the gland, which is marked in a cellular region in this one. So all the colors indicate the connected cellular regions that the software identified. So say in this in situ one, we have very large area here because it's identified the empty space as within the cellular area as well. But then when you go to the mix and the solid, we can see that the cellular areas identified are much more fragmented, and so we have different shapes there. And in this last line, we are showing the fiber extraction from this fiber network. And so each extracted fiber here is colored in a different way, just to show that we can identify individual fibers within this network. So if we look, say, inside here, we can see that we have different shape. We can identify the length of the fiber, if it is wavy, how thick it is, and we can count the number of fibers in these areas. And so with this fiber extraction, we can do a lot of detailed statistics on how the collagen is organized in the tissue. And one of the measurements, which is an interesting one to just extend that first idea of collagen organization in the tissue, is to measure the angle of orientation of each fiber. And so in this image, I'm showing as a color map here, the color indicates the angle of the fiber in the tissue. And we can see that in the normal tissue, it is a pretty colorful image because we have fiber oriented randomly in all directions. But with the cancer ones, we can see, say, for this one, we have a much well-aligned, thick area of fibers around this big tissue, this big cellular area grown, which is with the expand of the cancer. And so that fiber orientation, this one, again, when you get in the mixed carcinoma, where the collagen fiber is spreading to many different regions as well, we have a colorful image, but with identified regions of well-aligned collagens. And in the solid one, we lose a lot of the collagen within the tumor because it's mainly cells. And then it gets to be more colorful again, but in a different pattern from the normal one. So those were the ideas of features that we can extract from these images. Oh, sorry, I want to go back a little bit because I forgot to say one thing. And so in the cellular regions from these extracted networks, we can now evaluate which are the size of these areas, how is the density of cell as compared to the whole area. And so discussing with the pathologist, we came with 29 parameters, which they think are the important ones to classify the cancer when they are looking at the biopsy. And that includes then that cell density, area covered by cells, area covered by collagen. And then with this 29 matrix parameters, we can make a 29 dimensional space and look at all of them together in this using this machine learning technique, which is the linear discriminant analysis, which works roughly. Looking at this sketch here, is that we have a lot of parameters. I'm plotting it here in two-dimensional. So we have angles of presentation of this data that, depending on the axis, they are look quite mixed. But if we choose the right axis, we can project all this data along one direction that we can see them separated. So this is the idea of this technique, to search for axes to present the data in a way that they can separate better. So that's the idea of the linear discriminant analysis. So it's a supervised clustering method where we use the diagnostic, which is the diagnostic that was provided by the pathologist, as one of the labels for the images. And then what we do is that we separate all the images we acquired, which was on the 400 images, and we separate them in 6 sets, where we get 4 as training sets and 2 as just guess sets to test if we are selecting the data properly. And so there is those are machine learned techniques where we use this k-fold stratified validation method. So we train the software with part of the data, and then we use the second part of the data to see if the software is getting the result correct. And so looking at the histological type separations of the canine mammary cancer, what we get is this separation here. So those are the axes, which are the ones, according to the linear discriminant analysis, that projects the data in a more separate way. And the colors here are showing the normal, in situ, mixed, and solid carcinoma from before. And so we can see that the healthy tissue is really well separated from the cancer tissue. We certainly can see that all this corner part of the map is plotting all each dot is one image on the histological biopsies. And the full dots are the set that used as training set, and the crosses are the position guessed by the software with the unknown ones. And so we can see that the crosses are all positioned here, especially for the healthy one as compared with the cancer ones. And we can separate quite well also the very aggressive ones, solid tumors in this corner. But the mixed and in situ is somewhere overlapped here. So there's cyan and yellow is not very well overlapped. The star here marks the center of the set of data, and the ellipses is one standard deviation of the data. So we can see that we can separate quite well the extremes, but this middle part is still a little bit mixed. When we look at the cross-validation score, which is how well positioned the unknown data, the crosses here, are in terms of the number of features, we can see that we can achieve separation of the order of 6%, 60%. And the main problem is the separation of this in situ and mixed cancer. So that's where we got to this stage with this mammary cancer. And then we did another set of data looking for 445 female dogs with 836 biopsy regions, but not just looking at the details of the image, trying to correlate that with the prognostics, because they were done in just archive histological biopsies, and that the dogs were followed by 4 years. And so looking at the percent of survival of the dogs, those are slightly different types of classification of cancers, is carcinoma and mixed tumor, carcinosarcoma, micropapillary, and a solid carcinoma. And so that's the survival curve, looking at the diagnosis given by the pathologist at the time that the cancer were identified. And of course, the most aggressive ones were showing very short living time, but it was difficult to separate this more aggressive one. The survival curves are quite mixed. And then we looked at the details of the collagen fibers, and instead of separating the laminas by the histological subtypes, we are separating them by the collagen fiber length. And so we could correlate that there was a fiber length which was a critical point, that below that we have short-lived, well, the animal was living less, and above that there was a better prognosis. And so we can correlate the survival of the animal with the collagen fiber length. And we can see that we can separate all the animals with one cutoff, that below that it's a worse cancer because it was survival was small, and above that it was a better prognosis. So that is one first indication of correlation with clinical data, but it is a very pilot results yet, and so we have to carry on to see how that can be used in a more practical way. So just to summarize, in this canine mammary cancer, we identified that the imaging procedures allow good characterization of the tissue, and that the software, which is an automated images, provide quantitative parameters that can be of use to separate the cancer. And so in the classification between cancer and healthy tissue, we can achieve an accuracy of around 90%. For the different histological types, we can achieve a classification of about 60%. So it still needs to be quite well improved. And we observed a little bit of correlation with the animal survival with the collagen fiber length. And now I'm going to show just a little bit more of the results we obtained with prostate cancer. That's human prostate cancer. And now I'm going to say a little bit more about the classification patterns, which is based on these Gleason features, which classifies the tissue in different 5 grades. There are the types of pattern 3, 4, and 5. And pattern 1 and 2 is considered healthy still. and pattern 3 to 5 are the ones that are considered cancer. And the more aggressive one are the number 5. And actually, the diagnosis is given by a score, which is adding some patterns, because we have the final diagnosis is a sum of the most 2 common patterns. So the pathologists inspect the whole region of cancer that were collected by biopsies, and they give a classification which is 3 plus 3, 3 plus 4, 4 plus 3, and so on, where the first number is the most common pattern in the biopsy, and the second number is the second most common pattern. And so it is different to say that you have 3 plus 4 or 4 plus 3, because 4 is more aggressive than 3. But it's a very difficult classification. And so my colleague there, which is the pathologist, which is specialized in prostate cancer, says that it's really very hard to separate between these two. And then very recently, there was a review in clinical oncology saying that it's really a very big practical problems, because they went through 28 experts looking at the same biopsy, and there was an agreement of just 21.4%. And so it's really a challenging work for the pathologists to really give precise diagnostics, especially for this mixed, more intermediate type of cancer. But that is very important because it changes a lot the treatments and the prognostics. And so I think that's the major open question on this prostate cancer diagnosis from histological point of size, from the histological point of view. Because of course there are also the immunohistochemics to help that, but the other biological marks on the tissue, but they are still fighting to see how to improve these diagnostics. And so we did the same procedure as before. Those are, we studied 120 patients that were under cancer treatment in the hospital. And we had the safety procedures and issues with the human tissue there. All the patients are allowed that the tissue could be used for imaging research. And we could image 368 regions covering all the Gleason patterns by the nonlinear imaging. And to do the measurement, what we had was the pathologist was marking on the biopsy where should we do the second harmonic and fluorescence imaging, but not telling us anything about the diagnosis beforehand. And so we just had the tissue and say, measure in this area, measure in this area. And then we did the measurement, we did all the image analysis and all the parameter extractions. And after that, we printed them, well, we analyzed them with the same linear discriminant analysis and plotted against the diagnosis given by the pathologist. And so that's what we get. The colors here are representing the Gleason patterns, 3 plus 3, and so on. And again, we can see that the blue region, which is this part of the graph, separates well from the more aggressive one. But this intermediate area here, which is the 3 plus 4, 4 plus 3, is quite well mixed. But it's a difficult task because we were plotting the data as compared with the pathologist diagnosis, which is also imprecise. And so that's now we are just trying to work. Well, let me show you. So when we do the cross-validation score accuracy with the imaging and software identification, we can get an accuracy of 45% as to match the pathologist diagnosis. And so we had just 2 pathologist diagnoses. And so that now we are at the moment saying, well, how should we proceed? Because we have uncertainties in all the types of the diagnosis. And one of the things that we are just working at the moment is try to get an extra parameter, which is the fiber details of the biopsy, which is not included in that Gleason pattern, because the Gleason pattern is all determined looking at the behavior of the cells and the cell organization, and it's not including the extracellular organization. And so what the pathologist, and we are also discussing that together, is how to merge the 2 ideas to try to maybe get an extra way of classification, and how to compare that with the clinical data, because we are following these patients now for more than 2 years. And so I think by next year, when it is the end of the PhD project, we hope we have at least 3 years of following of the patients, so we can add to the project what happened as the diagnosis was taken and 3 years after, and see if we can correlate that and see which parameters are more important to predict the prognosis. So I think I'm going to be finishing now, saying that in terms of the prostate, the multiphoton imaging allows good tissue characterization, and we hope that the images analysis can provide quantitative parameters to help the pathologists get more precise diagnosis. And now we are working to see if we can do with that in some more fresh tissue after we get some way of saying which is the most important parameter, so we don't need to do it in the HE images biopsies anymore. So it can be a faster way that we can just get the tissue cut as in the first image and do it without any processing in any other color and cutting in specific sample preparation, to be able to see if we can compare that with the patient prognosis. And so I think that's, I'm going to stop here. Thank you for your attention. And I want to acknowledge the support from the funding agencies that we have in Brazil as well. So thank you very much for your attention.
Question? Okay.
Very interesting, but I think I can continue to make a histological diagnosis of prostatic cancer until my retirement timing, because I think that the morphological diagnosis is still the standard to make a diagnosis of cancer. It's quite curious that in morphological diagnosis, for example, of prostatic cancer, the structure of the collagen is not considered at all by pathologists. The diagnosis of prostatic cancer is based on the structure of the glands and into the glands from the evidence of nucleoli, for example. Very important to distinguish a small gland, for example, in atrophic glands from the grade 3 of prostatic cancer. It's quite curious, and we use staining for collagen fibers, for example, in some other field of pathology, for example, in the hepatic biopsies to understand the stage of the chronic hepatitis. And maybe I'm going to do some staining for trichromic or Masson staining into the prostatic stroma, because the prostatic stroma is made by the smooth muscle cells and by collagen. I can think that the growth of a cancer in the prostate can perturbate the environment of the stroma, but at the moment, the gold standard of diagnosis is glands, morphology of the cells, and also immunohistochemistry. I think that the agreement of 26 point something between expert pathology is quite low, maybe for an expert pathologist, but in general, there are some types of lesions that are very difficult to understand and to distinguish.
Yeah, from what yeah, thank you for your consideration, because yeah, it's like that. The pathologists that work with us was saying the same thing, that it is well, the Gleason patterns is all based on the cells and glands, and but now some of this reactive stroma is starting to be looked at to see if it can add some information on this cell patterns diagnosis. And this 24% there, which is said in this review, is specifically about this 3+4 and 4+3. Certainly the more aggressive ones are much higher. I can't remember the numbers, but I was talking about this 3+4 and 4+3, which are the most difficult ones to be, from what I learned from him, it was the most difficult ones to pinpoint on the biopsies. But yeah, I think this data here is just a very crude beginning. And we hope that in some time it can get to add some more parameters to help you to say in a very difficult one to have some extra information to see if it is going to help with the diagnosis or not.
If I can give a little suggestions about is to compare some types of benign lesions that are mimickers of morphological mimickers of cancer. For example, the atrophic glands in prostatic tissue that are quite difficult, and maybe that also the stroma can be perturbed by this type of lesion, for example, mainly if there is inflammations, associated inflammations.
Oh yeah, okay, thank you.
So congratulations for your work. it's a quite naive question because I'm not an expert and I'm not a pathologist, but I'm just wondering if you are showing data that basically used an algorithm, a software you named, with a mixture, an approach that is a mixture between unsupervised and supervised information. So you feed your software with a number of information from pathologists and the number of tissue section was quite limited, 400 or 500, you said, in some of your work. So it's, in your opinion, again, I'm not expert, but adding more and more data, we can expect that this software or algorithm will give us important information also for clinical impact or use with an unsupervised approach, or we have to deal with supervised, that is information that we need to give. So kind of cells, stromal features, and so on and so on. So I don't know in the future.
Yeah, the number of images and areas here is certainly very small to be able to train a software. It was just the first project. But yeah, certainly if we add a lot of images, say in these statisticals, things more than 1,000 would be at least the better starting point. And so that's the idea, just to keep adding information, measuring more and more biopsies before with the supervision, with the diagnostics, and then to be able to try to guess and do it in an unsupervised way. But I think that's still quite a long way to go, because what we are doing at the moment is just trying to see which are the most important parameters to keep in the software to be able to get the better precision. And so as the pathologist was just saying that well, we are not looking for the type of cells, we are just identifying that how is the shape of this mass of cell, let's say. And so because if we try to do it in a resolution to have the cellular details, then we can image very small areas. So that's another point. We were trying to image as large as possible area that we can do with the microscope and still be able to identify the fibers and the cells to cover mostly the most part of the biopsies, because we have to do in the same biopsy, we have to make many images to be able to cover an area that, looking at the normal microscope by eye, you slide then the biopsy, and then we can cover much larger areas. So that is another problem to tackle in the experiment, because we need to be imaging very many areas. And so it's a number of images to be acquired that takes a little bit more time. But yeah, there are still quite a lot of practical questions to be able to do it in a completely unsupervised way. But I think the main idea is not just to do it completely unsupervised, but is to just to have an extra number or an extra parameter to be able to rely if you have doubts about some specific diagnosis. And so it can just be an extra tool to add to the normal visual inspection of the biopsy.
Okay. One more question, and after Professor Mantegazza, sei in linea. Thank you.
Yes, just can you hear me?
Yes, we can. Now there is Professor Magni, and after, if you can wait 1 minute.
Yes.
Thank you.
Sorry,
Thank you.
excuse me.
No.
I have a similar question that already give you, Professor Invernizzi, about is a curiosity. Have you tried a completely unsupervised statistical analysis? Because sometimes in a supervised way, you confound the algorithm. In unsupervised, you can let the algorithm to do the best it can do, using maybe some neuronal approaches which can learn better than us how to see difference and similarities. At least in our experience, sometimes algorithms are better than us.
Yeah, we tried a little bit, but then for that, I think we don't have enough images yet. But we tried also to use random forest, which is another completely unsupervised one, to try to characterize which were the parameters that could best separate the data. So we are working on that. But for that, I think the number of images that we have is still small. So we need some more measurements to be able to do that. Okay.
Professor Mantegazza, please.
Yes, thank you for the opportunity to ask a question. I have a curiosity concerning the training set, the test set, which is the criteria that you use to separate the two ensemble. Have you tried to modify the two set and see how stable, how robust is the results?
Yeah, for that, we do it randomly, and actually we do it 6 times randomly, dividing the data in 6 groups, and always getting 4 as training set and 2 as test set. And for all the tests we did randomly, it gives these results that I was showing here. And so the robustness of the separation is given by this cross-validation k-fold method that the graph that I showed.
Okay, so thank you to all. Thank you for the attention to this very interesting seminar. Thank you, Professor Paula. And see you next. Thank you.